7-8 Science, Lesson 4. You should read before viewing this presentation, please. So if you haven't, pause. And we're back. It's called Using Genetic Information. So obviously the objective is to talk about the two ways it can be used, basically positive and negative. There's more positive ways mentioned in this lesson. By being able to identify individuals' unique DNA traits, we can learn a lot about health and disease. We can also find people and connect them to unknown bodies and suspicious crime scenes.
You've probably heard of the human genome, the huge collection of genes inside each and every one of your cells. You probably also know that we've sequenced the human genome. But what does that actually mean? How do you sequence someone's genome? Let's back up a bit. What is a genome? Well, a genome is all the genes, plus some extra, that make up an organism. Genes are made up of DNA. And DNA is made up of long paired strands of A's, T's, C's, and G's. Your genome is the code that your cells use to know how to behave. Cells interacting together make tissues. Tissues cooperating with each other make organs. Organs cooperating with each other make an organism. You. So, you are who you are in large part because of your genome. The first human genome was sequenced 10 years ago and was no easy task. It took 2 decades to complete, required the effort of hundreds of scientists across dozens of countries, and cost over $3 billion. But someday, very soon, it will be possible to know the sequence of letters that make up your own personal genome, all in a matter of minutes, and for less than the cost of a pretty nice birthday present. How is that possible? Let's take a closer look. Knowing the sequence of the billions of letters that make up your genome is the goal of genome sequencing. A genome is both really, really big and very, very small. The individual letters of DNA, the A's, T's, G's, and C's, are only 8 or 10 atoms wide, and they're all packed together into a clump, like a ball of yarn. So, to get all that information out of that tiny space, scientists first have to break the long string of DNA down into smaller pieces. Each of these pieces is then separated in space and sequenced individually. But how? It's helpful to remember that DNA binds to other DNA if the sequences are the exact opposite of each other. A's bind to T's, and T's bind to A's. G's bind to C's, and C's to G's. If the ATGC sequence of 2 pieces of DNA are exact opposites, they stick together. Because the genome pieces are so very small, we need some way to increase the signal we can detect from each of the individual letters. In the most common method, scientists use enzymes to make thousands of copies of each genome piece. So we now have thousands of replicas of each of the genome pieces, all with the same sequence of A's, T's, G's, and C's. But we have to read them all somehow. To do this, we need to make a batch of special letters, each with a distinct color. A mixture of these special colored letters and enzymes are then added to the genome we're trying to read. At each spot on the genome, one of the special letters binds to its opposite letter. So we now have a double-stranded piece of DNA with a colorful spot at each letter. Scientists then take pictures of each snippet of genome. Seeing the order of the colors allows us to read the sequence. The sequences of each of these millions of pieces of DNA are stitched together using computer programs to create a complete sequence of the entire genome. This isn't the only way to read the letter sequences of pieces of DNA, but it's one of the most common. Of course, just reading the letters in the genome doesn't tell us much. It's kind of like looking through a book written in a language you don't speak. You can recognize all the letters, but still have no idea what's going on. So the next step is to decipher what the sequence means. How your genome and my genome are different. Interpreting the genes of the genome is the part scientists are still working on. While not every difference is consequential, the sum of these differences is responsible for differences in how we look, what we like, how we act, and even how likely we are to get sick or respond to specific medicines. Better understanding of how disparities between our genomes account for these differences is sure to change the way we think, not only about how doctors treat their patients, but also how we treat each other.
All right. That video did a nice job of reviewing that short section at the beginning of 153. That Human Genome Project was a huge deal. There were actually two different labs basically racing to complete the sequence of a complete set of DNA for a cell. And they were kind of doing it different ways. But anyway, that was technically completed in 2003. And they're continuing to try to read that now and interpret what does it mean. I think it's really going to mean a lot for the future of medical science. And it's an exciting area of science to pursue. Again, geneticists have a really unique, interesting job. And I like how the video pointed out it's both huge, the task that they're taking on, but in a sense, so small and microscopic, right? That we're talking about 6 atoms across is a nitrogen base on a double helix. How we use genetic information can be identified by green is good and red is bad. DNA from a person's cell can be broken down and then fragmented into little sections, which we call a DNA fingerprint. With that fingerprint, since it's unique, just like a fingerprint, unless you're an identical twin, we can identify a person just with their DNA. We know that this is this particular person, as well as tell whether two people are related. Now, in order to be able to identify a person, we would need to have their data in a database. So sometimes we might be limited unless we have that person to draw DNA from, or we drew the DNA from them maybe when they were deployed. So by taking the DNA samples from sailors and soldiers, because they could end up dying overseas or battling or dealing with different challenges, it's useful to have that DNA on record so that we can connect it to that person later if the body's found. Forensic science, or science that analyzes a crime scene for the most part, can use DNA found to tie the suspect to a crime scene. Again, you can't just have one source of DNA or else you can't use it. You need to have two, right? You need to be able to connect it to something to make any kind of meaning. So DNA at the crime scene is fine and dandy, but it means nothing unless you have a suspect with which you can draw DNA samples from and see if they connect. So just having DNA isn't always going to matter. You need to have the person to connect it to. This is the way I would understand it. Unfortunately, the more DNA and genetic information becomes readily available, just like any information, it's becoming so much more accessible in this day and age. It could mean your privacy is invaded because your DNA fingerprint tells so much about you. It says everything from your genes and your family history to even your lifestyle, right? A little bit. Ethics tells us what is right, what is wrong, what's fair, what's unfair. And it's not always a clear decision, but the study of ethics asks, can we do this? Is it moral? Is it right or is it wrong? Is it against what people would want, against humanity, the laws of humanity, and being moral, being good to people? The Genetic Information Non-Discrimination Act, as you can see right in the name of it, we will not discriminate based on people's genes. So an employer can't even ask for DNA testing, let alone take it and then say, I see that you have a higher probability to have this disorder, so we won't maybe have you around long enough to make this hiring worth it. That's illegal. So your privacy is protected, not just with genetic information, but with any of your medical history. Not just with doctors, but actually this is the case with teachers too. Anyone who's given access to your medical records is lawfully required to protect that information and not share it with anyone else unless they have the same vested interest in you. So if you didn't know, when you share your immunization records or specific health records, or if a school district analyzes your ability to be advanced a grade, or if you need extra help, or you need extra challenge because you're gifted, all that type of information about you is personal. And when I learn it and put it on file, it's sealed, protected. I can't even talk about it with anybody else except for other teachers and your caregivers, your parents in all your cases. That's how strictly people protect your medical and genetic information, because it's personal. It says a lot about you, and you have a right to privacy there. That's what I think is really interesting about the coronavirus outbreak, COVID-19. So many people having it published whether or not they have it, because it's helpful as we work through protecting ourselves from something we don't know much about. Really, that's a violation of people's medical privacy. So hopefully in every case, they have released that information for people to learn, or they're sharing it themselves. But if they were to tell somebody in confidence, hey, I tested positive for the COVID-19 virus, that person doesn't have a right to share it on social media. Keep that in mind. Last thing, I put a poll up on Google Classroom. It's written in the form of a yes or no question, but by adding your opinion, it'll display to everybody, and we'll see where do you all lie on this. It's not a right or wrong, but it's a yes or no question. Let's say you learn from a doctor that you have such and such, maybe hemophilia, right? It's a genetic X-linked, sex-linked trait. That means that your alleles have been identified by telling other people who share family history with you, and therefore have an increased genetic risk to also have hemophilia. It would be beneficial for them health-wise to know that. But should you be forced to tell or to publish that information to your entire family? Should your second cousin know all of your genetic disorders just in case they need to protect themselves against the same thing? Be curious what you think, yes or no, and if it allows you to add a class comment, I'm not sure. I'd love to hear your opinions too. Thank you for... Oh, let's not forget, I got review questions. Very, very fun. What is a genome? What is a genome? Did you catch it in the reading? Did you catch it in the video? Your genome is your full set, an organism's full set of DNA. Ethics. What are we talking about when we call something unethical? If it's unethical, then it's probably unfair or unjust, at least by whoever called it that. When you're looking at a pedigree, this might be helpful as you're working through your workbook, which is also due on Tuesday. At the bottom of 154, are you going to choose a circle, a square, what, when representing a girl? Females are represented in a pedigree with a circle shape. Males with a box, a square, basically. Now, if you fill in a square versus what if you just partially, like half color in that circle? What does that mean? For that particular trait being tracked by the pedigree, it means that they carry the trait, they're a carrier, but they aren't affected, they aren't experiencing the symptoms. It's not seen in the phenotype. All right, for Monday already, I know it's not a science day, but I do need you to fill out your Chapter 5 test. So hopefully you've taken a look at this presentation to be reminded of that, or you see that on the checklist, but you're going to want to be familiar with Lesson 4 so you can get those questions right. Because I'm seeing your parents on Monday dropping off, I'd like this as soon as possible so I can get it corrected for you. Make sure you fill in all the answers in this short lesson, Lesson 4. And also I will hopefully have a Google quiz posted very soon where you can answer a few questions. I'm going to make this one shorter because it's a shorter lesson. Thank you for your work today. I appreciate it. Work hard. We'll see you later.